Hello, everyone. This is Jamie McCarthy, Marketing Manager from LabConnect. Thank you all for attending today's webinar, 3 Keys to Successful Global Sample Processing, brought to you in partnership with Marcus Evans. Before we begin, I would like to review the webinar interface. At the bottom of your screen are several application widgets you can use. All the widgets are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can also expand your slide area or maximize it to full screen by clicking on the arrows in the top right corner. If you have any questions, please ask them through the Q&A widget. We'll answer as many questions possible during the webinar, but if a more detailed answer is needed or we run out of time, we'll follow up via an email after the webinar. A copy of today's slide deck and additional help materials are available in the resource list. You can download any resources or bookmark any links you may find useful. For the best viewing experience, we recommend using a wired internet connection and closing any programs or browser sessions running in the background. You can also find additional answers to some common technical issues located in the Help widget at the bottom of your screen. Now I'd like to introduce our presenters. Joining us today are Elizabeth Corey, Vice President of Scientific Affairs at LabConnect, and Jill Marino, Associate Director of Bioanalytical Services, also with LabConnect. Liz Corey leads our Scientific Operations and Technical Affairs teams. Liz brings more than 16 years of experience in conducting and overseeing scientific research in the areas of neuroscience, oncology, and immunology. Joe brings over 15 years of experience in the pharmaceutical industry, and his experience includes method development and validation, process development and quality control, and analytical life cycle management. Thank you, Liz and Joe, for bringing and sharing your expertise with us here. And now I will turn it over to you.
Thanks, Jamie, and thank you all for joining us. Sample processing is something that Joe and I get very passionate about, as it's been a large part of our careers thus far. That's why we're very excited to announce LabConnect's Global Sample Processing Network. LabConnect has identified industry-wide needs for high-quality and timely sample processing due to the new demands and new complexities that come along with the current clinical studies. By establishing LabConnect's Global Sample Processing Network, we're able to capitalize on our strategically located global laboratories to be points of service for PBMC isolations and also for DNA and RNA extractions. With the establishment of this network, we feel that it greatly enhances the feasibility of processing samples with shorter turnaround times and allows for more monitoring of quality of sample processing as well. Today, we're going to discuss 3 keys that we feel contribute to the success of global sample processing. These are going to include specimen collection and transport, consistency in analytical technique, and safe and reliable biostorage strategies.
Today we're going to talk about the 3 keys to success that Liz mentioned before in a sequential order, kind of going from the collection all the way through the storage of the samples. We're going to start off by looking at collection and shipping under controlled conditions. Ensuring proper collection and shipping of samples is imperative. With sample processing, there's a focus on viability of cells and also stability of different markers that may be of interest in a clinical trial. These all require timely shipping as well as proper shipping conditions. Three analytical variation is a major concern in sample processing. The chances of a successful isolation will be greatly impacted by proper or improper sample collection. I think many of us have experienced an improper collection arriving at the lab for processing that we're unable to process due to the collection tube type or the matrix that the sample is in. Therefore, collection supplies should be well labeled and well organized.
That's very true, Joe. That is a common issue that we see. As such, LabConnect really has had a lot of success with their kits. Their kits are very well organized. The tubes are very well labeled. They are laid out according to collection sequence as needed. So overall, they are found to be very user-friendly at the site and very much less prone to these types of errors.
Thanks, Liz. In addition to the collection tube types, shipping supplies and conditions are also another critical component to this process. Shippers must be validated to ensure that proper temperatures are maintained. Samples must be packed correctly to avoid damage or loss during shipping.
Turnaround times are always a major focus when it comes to sample processing due to sample stability and viability. So for example, with PBMCs, we tend to target a 24-hour turnaround time. However, with these newer, more complex studies, we have gotten requests for up to 6 to 8 hour turnaround times, which makes it very challenging. And then when you add international shipping to the process, this will add a whole nother layer of complexity. International shipping is prone to unforeseen challenges, such as infrastructure failures and, of course, delays in customs. So in order to minimize this risk, we would suggest partnering with a central lab that has access to regionally based laboratories. This is going to minimize shipping distances and duration, which in turn should lead to greater viability, sample stability when it arrives at the processing laboratory.
I agree here and want to also emphasize that you need to understand the stability of your analyte of interest to help inform your central lab decisions and where to place collections on a regional basis.
The ability to track your samples in real time is another highly desirable option in this process. Tracking the movement of your samples from accession to receipt at any lab or storage facility in the world is critical to your peace of mind. The benefits of this tracking include enhanced security and integrity of samples, increased control over shipping logistics and costs, easy report generation, easy retrieval, and also allows for a chain of custody trail. LabConnect addresses this challenge by using our digital pen technology and our SampleGistics platform. This enables us to know where samplers are at all times.
In this slide, we wrap up the first key to success in global sample processing, collection and shipping. Thus far, we focused on well-organized sample collection kits with clear instructions, shipping time requirements, and selection of time-dependent regional facilities. And finally, we'll focus on selecting the proper shipping partner. Appropriate sample management and regulated bioanalysis is crucial. If samples are compromised at any stage prior to analysis, study results may be affected. 2 questions to ask when you're considering which carrier to use are, can your carrier offer temperature-controlled transport of your specimens? And do they know the local geography and politics well enough to efficiently retrieve samples from rural areas and negotiate through customs? LabConnect has addressed these questions and partnered with Martin, an expert in international shipping of time-sensitive materials.
So now we're going to move into the second key to success. The second key to success is identifying proper global laboratories for your particular studies and vetting these laboratories to determine their quality and capabilities. Previously, we discussed lab selection based on time and logistical challenges. But now we're going to focus on finding the right laboratories that can deliver high-quality processing by vetting certain parameters such as lab quality systems, lab instrumentation and capabilities, training processes, analyst qualification criteria, and the strategy that the lab has in place to monitor performance over time. Your central lab should have the experience necessary to vet partner laboratories for their abilities in sample processing. Focusing on quality systems and capacity capabilities, you will want to consider a few things. First, SOPs. Does the laboratory have SOPs for each method, and are they up to date? You also want to consider if analysts in that laboratory are qualified on all of the methods, and is the documentation in place? Does the laboratory have adequate instrumentation to support the demands of your trial? Are the instruments up to date on preventative maintenance and calibration documentation? You'll also want to consider if the laboratory has adequate capacity for your sample volume demand in terms of lab space and also in qualified analysts. And then you want to consider if the lab is available for weekend and off-hour processing to support Friday collections or delays in shipping that may occur.
In addition to quality and capacity considerations, technical considerations will likely also come into play. In the instance when a comparable method is required to be run globally, we would need to ask, does the processing lab have the ability to change methods and potentially qualify and perform additional qualification activities? Consistency in processing is a critical factor. All labs will have personnel attrition. However, a robust training and qualification program will ensure processing continuity. Having globally standardized protocols ensures uniformity in training and consistent quality expectations.
Thanks, Joe. Analyst qualification is highly important, and LabConnect has addressed this challenge by implementing a performance assessment for all the labs in the LabConnect Global Sample Processing Network. What this assessment does is it follows a training model of observations, side-by-side processing, solo processing, and also has a very clearly defined acceptance criteria for qualification of all analysts.
And then following training qualification, also having a mechanism to routinely track analyst performance is essential. And we'll take a look at that in our next slide with performance monitoring. The ability to review global data in a centralized, consistent format is essential to both monitoring analyst and process performance. Great analytes for process monitoring in a PBMC process are overall cellular recoveries and viability. In a DNA process, concentration and purity are good metrics. If monitoring parameters start trending in a concerning direction, immediate evaluation of processing materials, laboratory instruments, and analysts should be performed. It is also important to understand clinical expectations, as performance monitoring parameters can be impacted by different sample types. It is also recommended that samples are tested for their intended purpose early in the trial to ensure quality and address any potential issues. You do not want to get to the end of a study before testing downstream analytics.
That's a great point, Joe. LabConnect has put in place a monitoring system in their global network where the laboratory data will come back into a centralized database and personnel will be dedicated to reviewing that data. So in that case, we are able to ensure that performance is adequate and at high quality at all times. And then on the flip side, if there is an issue, we are able to address that in a very timely manner to avoid any further downstream issues. So just to wrap up this section, we have touched on the importance of quality systems and capacity, the importance of having a robust analyst qualification program in place, and then also the importance of continuous performance monitoring. Now we're going to move into our final key to success. The third and final key to success is ensuring proper storage for all samples and data after the samples are processed. It's critical that once processed, samples remain at the appropriate temperatures for storage. If samples are being frozen for long-term storage, consideration of storage space and location should be established before the samples are actually processed. In this section, we will evaluate backup requirements, space and freezer requirements, and security considerations. When considering your biostorage accommodations, you will want to evaluate the following. Are the freezers fully validated and mapped? Are there backup generators? Is there redundancy in HVAC systems to protect your samples long term?
Liz, it's a really good point to have the backup freezers proactively mapped. According to Murphy's Law, freezers tend to go down in the middle of the night with the primary point of contact out of office. So a clear plan will really help in that type of situation.
I'm a big believer in Murphy's Law. So another point to make and to consider is to always have your primary and backup samples in different freezers should one go down.
Freezer space and monitoring conditions are often overlooked as critical factors. Prior to choosing a biorepository, your sample stability and storage requirements should be well understood. Verifying freezer space, temperature ranges capabilities, and overall capacity is important. In addition, understanding continuity plans for equipment failures will help ensure success. There are many unforeseen issues that could put your samples at risk. Choose a lab with good security and one that has plans in place for emergency situations. The lab should have bag access to storage locations. Equipment should be on alarms and continuously monitored for temperature excursions, and the overall building should be secure with a security system.
Prompt retrieval and shipment of samples is very important. The timing of retrieval and shipment will vary with every trial. Therefore, a clear understanding of accessioning and an upfront planning of sample retrieval requirements should be established. Streamlined accessioning and inventory management systems should be solidly in place. In this webinar, we have touched on 3 critical factors to achieving successful global processing. They are collection and shipping, identifying the proper laboratories for your studies, and storage conditions. What we've learned is that having strategically located processing labs and standardized global processes will result in shorter shipping times, quicker turnaround times, and enhances the possibilities of increased viability and recoveries of samples being processed. In addition, well-designed collection kits, validated shipping materials, standardized analyst qualification, and centralized performance monitoring will all contribute to the success of sample processing and help to ensure consistent and quality performance across laboratories.
In addition, proper storage is also essential to ensuring your samples are secure. Identifying redundancy in these systems will further ensure there is no loss or compromised samples. Overall, be thorough in choosing a central lab and your sample processing partners. Thank you for joining us today, and we hope this was helpful.
Thank you, Liz and Joe, for sharing your insights. We do have some time for questions, so if anyone has any, please make sure that you send your questions to us, and we'll answer those right now in real time. Also, we'll make sure to get back to you via email after the webinar if you have some questions that we do not have time to answer. Thank you so much. Thank you, Liz and Joe, for that webinar and all that information. Very valuable. Now we have time for some questions. Our first question is, what type of parameters do you evaluate for analyst qualification?
So I think
Joe,
this is yeah, I can take this one, Liz. I think this is a good question. Sample processing is a pretty broad topic, so qualifications could look very different depending on what process you are looking at. So in some cases, if a process as simple as aliquoting serum, the qualification protocol could be fairly simple and not too complex, maybe just review of protocols and some test aliquoting, universal precautions. However, we could go to more complex processing, like bone marrow mononuclear cell isolations, and at that point, the qualification would look a lot more complex. In a way, one example of our PBMC isolation, what we do at Lab Connect is we follow the model, as mentioned in the presentation, of observation, side-by-side, and solo, where the new analyst will observe the process by a trainer and then work side-by-side with the trainer and then finally execute the process on their own. And every step of this process has clear defined acceptance criteria, so we know that the analyst is qualified. And we really look at both cellular viability and the recoveries. In addition, we actually also put in a post-thaw viability requirement just to ensure that the cells coming out of thaw also look pretty good.
Okay, great.
Thank you. I was just going to add to that real quick. So we also have, for our DNA and RNA extractions as well, qualification processes in place with our laboratories. And with those, we look at concentration and purity outcomes. So again, just to add to what Joe said, we do ensure that the laboratories are running, multiple analysts, multiple runs of whatever process they are doing, just to ensure that everyone is qualified.
Okay, moving on to question 2. What is the toughest sample processing situation Lab Connect has encountered, and how is it resolved?
Oh, that's an interesting question. I think when you're working in clinical trials, anything can happen, right? So the clinical state of the donor can really impact the sample type. So really being able to be flexible in your process and be ready for anything is important in the processing laboratory. I'd say one of our most common situations that we see is under collections. So again, that clinical state can impact, the ability to actually get a full collection from a subject. So being adaptable with your process and being able to adapt to different sample volumes will be important. And, also understanding when you can adapt and where you can adapt. In the end, I think in this situation like this, proper documentation and making sure that, we're communicating back to the collection sites and back to the sponsors so they understand, what's happening, and understanding the situation on these subjects is also important. In terms of the toughest sample processing issue, I think we have received at one point from a leukemia subject a PBMC isolation that was just huge, and it was going to require 40 samples to be frozen down. So being in a process like this, once you get into freezing media, cells can tend to die. So our team quickly adapted and put multiple people in the hood together to get those samples frozen down as quickly as possible. So again, it's really the clinical states that sometimes dictate these challenges, and just being ready with a laboratory that understands the situation and can adapt is very important.
Fantastic. Thanks for that comprehensive answer. Moving on to question 3. The question is, what makes Lab Connect's offering of a global sample processing network unique?
I'll take that one. So I think, there's a few things that really kind of make this offering a little bit unique compared to other situations. And I know, a lot of clients have their own laboratories that they go to for their routine kind of sample isolations or sample processing and things like that, which is great. And what we're trying to do is really capitalize on our network of laboratories that we have globally now that we know are quality laboratories and that are laboratories that we have already built very strong relationships with. And so we thought, this is going to be very helpful in being able to accommodate, quicker turnaround times and, less loss of samples and any kind of errors in accessioning samples and, drawing samples and having them processed. So I think that's a big aspect of it is just our current network of laboratories that we can capitalize on to really provide this service. Again, having relationships with these labs is a big deal to begin with, and then we're also able to provide more continuous oversight of these labs as things progress. Another thing that we do is we do kind of the standardized qualification, as Joe had mentioned, I think a few questions back. We have, standard qualifications for analysts depending on what processes they're going to be performing in those laboratories. And all of that qualification data comes back to us, so we're able to review that. And we're able to monitor when new analysts come on board. So that is a big, I think, a kind of a unique offering too at this point. And then finally, I think the general consistent monitoring that we are providing of the quality of the processing that is coming out of these laboratories. So as mentioned in the webinar, the lab will process samples and that general data will come back to us into a centralized database. And we will be having, we will have someone reviewing that data for quality. So we can see sort of, the consistency of the processing that is coming out of each laboratory. And also, if there are any issues, we're able to address those very quickly. So I think that extra kind of layer of monitoring that we can provide so clients don't really have to worry about it as much is definitely unique to what we're seeing in the industry right now.
Great. We have a question from one of our viewers as our next question. The question is, we receive samples from sites participating in clinical trials. In some cases, the tubes do not meet the requirements as agreed. What measures does LabConnect take as a CRO to avoid this and guarantee quality of samples?
Liz, maybe I can start off with this one. So I'm thinking that it sounds like this is a tube question, and in terms of the quality of the tubes or maybe the consistency of the collection into different tube types, I think one of the unique things about LabConnect is we do build these custom kits. So across the global network of collection sites, they will all have a uniform kit built. So you aren't worried about incorrect tubes being placed into the kits, and so collections should be consistent. So I think that is probably one of our primary ways that we handle, not having tubes that don't meet or having tubes that do not meet the requirements.
I think that's exactly right. And we, one of the things that LabConnect does is build our kits by hand, and kind of the company and that team specifically really prides themselves on building kits that are very user friendly. So we definitely see less mistakes in this type of area.
Great, thanks for answering that. Our next question is, how do you deal with samples stuck in customs? What can negotiating entail?
Well, that's always a tricky one. I can get this started here. I think, one of the things that we do is that we provide the documentation to customs in advance of the package arriving. So this will allow then, review and sort of if there's any issues, they're able to be resolved before the package arrives. And I think that's been a big factor in kind of avoiding longer-term issues in customs. We also work to ensure priority sorting through our couriers. So our packages are always kind of among the first in line to be inspected and cleared through customs. We also really, again, it's a lot about relationships as well. And, we do, work closely with Markin and between the companies, really have close relationships with custom agencies globally. So, this is something that is common in our logistics department, unfortunately. But we've certainly found ways around it. But I think, for other clients or things like that, when you run into that issue, I do think that a lot of the success that we see are because of the ongoing relationships that we do have with these custom agencies.
And I'll hop in here just to agree with Liz for sure that the pre-clearing documents is really important. Just as many things that you can do up front to try to pull through those samples through customs is important. And then on the other end, also having a shipping courier that understands the sample types too. Sometimes samples are shipped frozen or at room temperature, but making sure that that courier understands that when samples do get stuck, what actions do they need to take to ensure integrity of the samples is important when things happen in customs.
Yep, agree. Thanks, Joe.
Thanks, Joe and Liz. We have another question from one of our viewers, and the question is, can you further describe the digital pen that's used to track samples in real time? For example, how is it set up with a sponsor, and what do the sites need to do so the sample flow is tracked on the pen?
I'll start this one off. So the digital pen is something that the sites will have, and the uniqueness of the pen is such that when samples come in, sites will be able to write like any other pen on these digital requisition forms. And they'll accession the samples on these forms, so all the sample information will be written out on this form, just like any other form or piece of paper. When the pen is docked, all the information that was written down on those forms goes into our centralized database. So we're able to see all the information about that sample in real time. And those forms are reviewed once they hit our database. And then any kind of error, discrepancies, or any questions that we might have to go back to the site for, just to make sure we have the accurate data for those samples, we do that immediately. So you can always see, so then sponsors can log into our systems and always see where their samples are at any given time. So there's some instances where samples come, they get accessioned at the site and then they're sent to Lab Connect or wherever it may be. And then there's other instances where they're sent to specialty laboratories for testing and things like that. The pen enables us to be able to see wherever those samples are at any given time, which I think it really does offer a lot of peace of mind to our clients.
Okay, great. We also have a follow-up question, and the follow-up question is, you mentioned that Lab Connect hand builds kits. What's the QC process for those kits to ensure they're built correctly?
Well, I'll take that one. So I would have to honestly get you the exact QC process that they do, but they absolutely hand build the kit, and I know it is review of that kit build by another person on that team. So it's very, it's something that they take a lot of pride in, and Lab Connect takes a lot of pride in, and I can get you the details of the process, but I do know for sure that once the kit is built, there will be a review by another person on that team just to make sure that all the boxes are checked.
Fantastic. Thanks, Liz. That's about all the time we have for questions today. And I would like to go ahead and thank you very much for your time and attention. Thanks to all our viewers who have joined us today. And I would like to remind everybody to keep an eye out for an email within the next couple of hours that will have links to download today's material. We'd also like your feedback, and so please take a minute to answer a brief survey that will pop up on your screen at the end of the session. On behalf of Lab Connect and Marcus Evans, thank you for joining us, and we hope to see you again soon.